Well, good morning, and welcome back to day 3 of our omics analysis marathon. Viewers seem a little thinner on the ground this morning. Maybe they're having trouble finding parking. Maybe we've worn them out. And I'm assuming that those of you who are still here aren't just coming for the coffee and pastries. But we're going to get started. And our subject today is classification. I'm going to make a sort of couple preliminary remarks. First is that I've obviously prepared an HTML file from a Markdown file today instead of a PDF file from something else. Again, if you go to the Box website, Coombs, Lecture 09, the R Markdown source as well as the HTML file are there if you want to read through the source at some point to see how this was done. I will also mention that here's the table of contents. You will see the block under Run Some Classifiers that I'm going to try to talk about at least 10 different methods to build classifiers. So you might suspect that I'm not going to go into full detail on all of them. But I do want you to understand something about how they work. That's part of the point of this. And I also want to put this into context because in the applications of omics scale data sets, there are basically 3 kinds of problems we attack. And we've been working through them in order of increasing difficulty. The first of them usually goes by the name of class comparison. You have 2 groups, sometimes more groups. You know what the groups are, and the question is, what's different between them? And so the talks that Greg Rampala and Maciej Petrak gave yesterday on differential expression, that's a primary tool for attacking the class comparison problem. The second kind of question is class discovery. We don't know the labels. We want to discover what structure is present in a data set. That's the clustering talk that I gave yesterday. And the hardest and the kind of problem that everybody really wants to answer typically is class prediction. We know labels, but what we want to do is to learn from some sort of training data set how to make accurate predictions on the next data set. See, making predictions is easy. Making correct predictions is hard. So that's what we're going to try to learn how to do today. Talk about classification as a tool for predicting what's going to happen in the future. Let's see if I can. So the first thing that I'm going to do, we're going to ignore some of the beginning of this, although it's here, is that I changed what I wanted to do in this talk between Tuesday and today. And so that whole list of packages that I had you install didn't include a couple more that I'm going to need for this lecture. So if you're actually going to run through it on your own from the source, you need to install rpart.plot, bimodal index, ROCR, and E1071 if they're not already installed. So there's code up there to do that. That's boring stuff. I tend to find the colors more interesting. One of the packages that some people were unable to install, so I'm working around, is called Polychrome. I'm going to use R Color Brewer, which is the older package in R for building color palettes. One might guess that the brewer is sort of a pun of you brew up the colors. Actually, it's Carol Brewer who designed this plan for colors, so it's actually named after her. And this is one of the palettes there that we're going to use some of for today. The part that I hand-coded from Polychrome is this color scheme that I'm going to use. And Polychrome is designed to give you colors that you can more easily tell apart. We'll see if it actually works. And now we're going to actually start on the core of what we want to do today. I want to illustrate all of these classification methods. And I'm going to do it with carefully simulated data. Because with real data, it's a little harder to tell. I'm going to put data just in 2 dimensions so we understand it completely. We understand where it came from. And part of the goal is to actually simulate something that's complex enough to actually challenge the classification algorithms that we're looking at. What people usually do when they do these simulations is they simulate 2 groups, one each from some multivariate normal distribution, and it's really easy to tell them apart. Well, classification is hard, so I want to make sure that the example I simulate is relatively hard. So I'm going to talk a little bit about how I'm doing that as well. So there's a whole big, ugly chunk of R code here. right? By now, you're supposed to not be scared of R code. Mathematical equations, maybe you can still be scared of, but R code is now just routine. Okay? So what is it that I'm doing here, right, as we work through this? Okay? There's a key step here, this set.seed. Since I'm simulating this from some pseudo-random number generator in R, I want to make sure that the simulations are reproducible. Set.seed puts the random number generator in a consistent state, so if I rerun this program again, this script again, it will simulate exactly the same data. We pointed out that if we picked normal numbers and we didn't do that in one of the first talks I gave, we'll get different random numbers each time. If you set the seed, you'll get the same random numbers again, so you can make your script reproducible. And then I'm choosing a whole bunch of random numbers. Let's see. The heart of this, buried way down here, is I'm choosing means and standard deviations and then simulating from a multivariate normal distribution. And the magic thing is that I'm doing this 8 times. That's what you need to know about this block. And we can see from the plot command down there is that as soon as I go down a little bit in here, we're going to see a plot. So this is my simulated data. So it actually comes from 8 groups, each with a different color here. We know how to put legends on, so we see what they are. And we see that they're sort of spread all over the place, because we've changed what the mean is. We've changed the correlations between them, the standard deviations. So I'm generating stuff from 8 groups of stuff. And I'm not going to actually try to predict all 8 groups. I'm going to arbitrarily, not quite arbitrarily, because I looked at this and made a choice. I'm going to put 4 of these groups and call them A, and the other 4 and call them B. Question?
What is the function C bind or R bind?
Which function?
So there's the code from Pocket that it says R bind. It's like sim2d do call R bind.
So that's a good question. So R bind is if I have 2 matrices, I'm going to glue them together by rows. So R bind is to glue the rows of one onto the bottom of the other. Okay? So there's some subtle R stuff in here because do call is sort of a magic way to do things. It's going to apply this function to this list of stuff. So if you've built a list and you want to use this as argument, so R bind will take a list of matrices and bind them together. As long as they have the same number of columns, they'll stick them together as new rows. And if you actually have named each one of them separately, you could say R bind of A, comma, B, comma, C, etc. But here, I built a list by running this sapply function. So temp is a list. And so if I want to do that, either I have to manually name each piece of that and write the code, or I can use this do call trick to apply this function with this as the list of arguments. Okay? So there's some subtleties there. As I said, this is the data. And then I've made this manual hard-coded map. Mappers is which groups are called A's and B's. And this is the data set that I'm going to learn how to classify. And as I said, by simulating from this mixture of multivariate normal distributions, it's not obvious how to separate the blue area from the orange area. And that's good, because that means we're going to actually be able to see how our various classification methods respond in terms of trying to do that, how they figure out where to draw the boundaries between the orange area and the blue area. Because that's what classification ultimately is going to do, right? Some things it's going to label orange in the future, some things it's going to label blue. How does it do that? Where does it draw the boundaries between one and the other? Okay. And so I'm also going to build a test set from the same kind of data so that I can test how this actually works on new data as well. So this is another simulation. To begin with, right, the clever point is my test set is a grid that covers that whole region. So I'm going to tell you for each point, I go up here, for each point inside that square, inside the x and y axes, I'm going to make a prediction as to whether it should be orange or blue. Okay? All right, so how am I going to do this? Well, first I'm going to look at what truth is. I know exactly the distributions that things came from. There were these 8 groups. Each group is coming from a multivariate normal distribution, which I'm pretending you know what it is. All right? Well, what does that mean? It's in this case, 2 dimensions. What makes it multivariate normal basically is there's a center someplace that's known. There's a distribution that I'm simulating from. And there's a 2-dimensional picture of what the correlation structure is and what the standard deviations are. So you can think of each distribution as coming from a set of ovals. There's a center, and that's maybe the 20% and 30% and 40%, whatever. There's some direction, right? As in principal components, there's 2 primary directions, a major axis and a minor axis. Those proportions stay the same. And the probability of being from this distribution decreases as you get further out. And it decreases not necessarily the same amount in the different directions. Other distributions, this is tilted, stretched. So there's 8 of these. And so what I can do is for each point in the plane, I can see which of those 8 pieces has the highest probability of being, let's see if I want to say this. For each point in the plane, I can tell you the highest probability among those 8 distributions for it coming from one of that one. So I can pick what's called the maximum likelihood model for each point in the plane, which I can do from the actual data that I simulated from. So not from the data, but from the known model. And so if I do that, this is how the plane would be colored in terms of the 8 groups. Sort of unusual. You can sort of see these ovals getting squished over by other things because when you get to the center of each one, it has a stronger influence. And when they overlap, weird things happen. There's that purple one that sort of goes across the middle that is obscured by both the darker blue and the green, because those are higher probability in those regions, but the purple one spreads out, and there's some regions where it's the maximum probability, the maximum likelihood. So again, it's a complicated simulation. Each of the 8 colors dominates some part of the plane. Some parts are most likely to be there. So that, in essence, if I was trying to get all 8 classes out, that's the true maximum likelihood choice as to how you colored the points in the plane. On the other hand, we want to see what happens if we turn this into 2 groups, which is what we did. And so we can do something simple. Here, this just says, of those 8 things, which one has the maximum probability, and then turns that into either blue or orange the same way I did this. So this is one approximation. I'm telling you there's a lie here, so it's an approximation of what we think the maximum likelihood would be, what the underlying colors of the planes are. So it tells me for each point in the plane what's more likely to give rise to that. Is it the orange group or the blue group? And you can see it's sort of complicated here. The difficulty here is that I've added things up by just taking the maximum of the 8 pieces. And what I should really do is add them up in a probability sense. That's what this does. What I really want you to think about, I want you to burn this picture into your brain, because this is truth. For the data I've simulated, this is the true answer, and that underlying orange and blue background to the plane is how things should be predicted, because that's the maximum likelihood of the A-B groups as to what gives rise to the data. Now, there's an important thing to note here. Even the maximum likelihood model based on truth would predict some of the data points incorrectly. That's part of my building a really challenging example to learn classifiers on. Right? There are blue points that are clearly in the orange region. Rare things happen. Right? So that's one of the things to keep track of here. Right? We can walk over here and we see there's blue stuff there and there. There's orange stuff here and here that is going to get predicted to be the wrong color, because that background color is what the maximum likelihood model based on truth would predict. So it's challenging in that way. The other thing, of course, is that the boundaries between the orange and the blue region are quite complex. They're not simple-minded straight lines or pieces of straight lines. Right? There's an orange island in the middle of the blue river It doesn't have any data points there, but if there was a data point there, we would expect it to be orange. Okay? And we actually see both banks of the river. There's that orange stuff down in the bottom right-hand corner, again, where there's no data there. If there were points there, the model says they would be more likely to come from the orange group than the blue group. Okay? So I think I have succeeded in simulating a data set that should be challenging to try to figure out how to model and how to classify new points. Okay, so now we're going to try to actually see what different classifiers do on this kind of data. So the first model I want to talk about, at least briefly, is called this linear discriminant analysis. And there's various ways to think about it. The goal here is to draw a straight line that does the best job of separating, in this case, the blue data points from the orange data points. So the classifiers we have now don't know truth. They just have the data that's been simulated. They're trying to learn truth out of that data. Okay? Because we knew how we simulated it, we knew truth, that was that last picture. Okay? So linear discriminant analysis, the first thing is going to draw a line. Well, if it's in 3 dimensions, it's going to draw a plane. If it's in more than 3 dimensions, we run out of names for that. We call them hyperplanes. But they're straight, they're flat. Just linear equations that separate the two regions. Okay? And there's a sense in which linear discriminant analysis is a generalization of ideas from the t-test to higher dimensions. The t-test, how do you compute a t-test? You take differences in means, you divide by some number computed out of the sample size, and you divide by the standard deviation. Well, here the underlying idea is you're fitting multivariate models. The standard deviation is actually a covariance matrix because it has to account for correlations between different variables as well as the different standard deviations. And so one of the difficulties in applying this in at least some cases is you have to find the inverse of that standard deviation matrix, that covariance matrix. And that actually has an implication in omics because if you have more variables than data points, if you have more genes than samples, that matrix isn't invertible by definition. So it raises some challenges in applications. But here we just have a 2-dimensional space. We're going to be able to invert it. We can work out what the linear discriminant analysis is going to give us. And of course, we're not actually going to compute it manually. We're going to use a function in R called LDA, for linear discriminant analysis. You feed in the data and say that the categories in the data are a function of everything else. That's this shorthand in R. We've got a category someplace, and we've got however other variables there are. This is a function of, in a statistical model sense, and dot means everything. Okay, so I don't have to write down the names of all the variables. And I'm using my simulated data. And so that line fits a model, which we've cleverly called a model. And you make predictions of the model on the new data, and the new data I'm using is the grid of points behind my region in which I'm making the predictions. And in this case, it makes them numeric, and we're going to turn them into a class, and we're going to see how well this works. So again, we have the same data points plotted here, but what we've now done is the background is the predictions from the linear discriminant model. So you can see the line that separates things. Given what we know about the actual shape of this, lines aren't going to do a fantastic job, right? Clearly, it's going to get some things wrong. There are a lot of orange ones over in the blue region. There are a lot of blue ones over in the orange region because the actual border is some windy curve. Okay? But this is the best you can do, in a sense, with linear discriminant analysis. It's also worth looking at how does this work on the training data. Now, one of the lessons I also want you to learn about this is that if you just try to estimate performance of a classifier on the training data, it always looks much better than it will look on test data. That's the best you can possibly do, is test your model on the data that you learned the model from. You can't ever do better than that on test data. If you do better than that on test data in a significant way, there's something wrong somewhere. Okay? So be suspicious of that. If somebody, on their training data is 80% accurate and on their test data is 97% accurate, there's something fishy going on. Okay? But that also means you actually need independent stuff to test on at some point. We'll look at that later. But even on the training data, because we've made a challenging example here, this linear model doesn't do great, right? It gets, well, we can work out what the percentage is here, 110 A's right, 93 B's right, but it gets a total of 89 wrong, right? So out of the not quite 300, 90 of them are wrong. That's a significant error rate on the training data, okay? And in fact, it only gets 69.5% right, okay? So I'm pleased that I actually came up with a simulation that makes sure that this doesn't work. Linear discriminant analysis here doesn't work. If your data doesn't have a nice linear separator, you're not going to get your best classifier out of a model that just draws straight lines, okay? All right. So now we're going to look, I know there's a math equation here some people are now scared off. Get over it. So we're going to look at a tool called logistic regression. So the underlying model is that what you want to model is the probability that some sample belongs to group A. And you use what's called a logistic transformation. You take that probability P and compute log of P over 1 minus P. Well, there's a very simple-minded reason for wanting to do that. Probabilities live between 0 and 1. And fitting linear models to do that does really complicated stuff because they extend way outside of there and they make predictions on probabilities that are 37, which isn't a good thing. So if you take this logistic transformation, instead of being between 0 and 1, now it goes from minus infinity to infinity, just like my linear predictors might. So in that sense, making this logistic transformation on the probabilities is reasonable. There's other reasons beyond that why we might want to do it. But this is what we're going to do. We're going to try to predict log of P over 1 minus P. And if you have that, you could solve back for what P is. It's pretty easy to invert that function. And we're going to write that as a linear function of whatever predictors we have. We often fit things with linear models. Sometimes that's because we think it's really the right model. Sometimes it's because it's convenient. It's really easy to fit linear models. There's nice, simple, deterministic algorithms that do it quickly. So you can at least get an answer. Then you have to assess whether the answer is right or not. That's what we're trying to do here. This gets fit in R using GLM. Greg Rampala talked a bunch about generalized linear models yesterday. That's the underlying tool to do this. It's a generalized linear model because of that logistic transformation of the thing we're actually trying to predict. Right? We're not just trying to predict P directly. We're trying to predict some transform of it. And so we do that, and it's called a binomial family, because it is. And here's the model. Right? It says that x and y, there's an intercept, there's an x and y term, because we have this two-dimensional data. And that's going to be used to make predictions. And so, as with many of these things, you fit a model. That's the line to fit the model. And then you make predictions on your new data. And this actually just gives probabilities out in the form that we're getting them. And I'm going to turn that into A's or B's so I can test how well it works and see what happens. And to see what happens, well, that linear part of the model, the fact that we're writing the probability as a linear function of the predictors, actually again turns out to divide the space with a straight line. So it's not going to do great. You have to look very carefully to note that it's not quite the same line that we got from linear discriminant analysis. Right? If you get a copy of this and scroll back up and you look very carefully, the line tilts just a little bit from the optimal line from the point of view of linear discriminant analysis. So it changes the predictions a little, but not very much. Right? We get similar numbers in terms of accuracy. We get a 69% accuracy, a 68% true positive rate, that's also known as the sensitivity, a 69.9% true negative rate, the specificity. Okay? So we've looked at two linear models. Those are sort of, globally trying to find linear models. We're now going to switch to a different kind of classifier that's motivation is completely different. So we're going to use something called K-nearest neighbors. So you get to choose K, how many nearest neighbors there are. And the idea is that you're not going to try to fit some nice globally shaped model. You're not going to insist on a line or some particular kind of curve. All predictions in this case are local. So if I get a test point that I want to make a prediction on, what you do is you find its nearest neighbors, however many you specified, and you let them vote, and majority vote wins. So it's a very democratic classifier. Depends then on how big a neighborhood you canvass. The smallest possible one is 1. Oh, and by the way, you always choose K to be odd, because otherwise you need some stupid way to break ties. If it's odd, there's no ties to break. So what happens to K is 1, right? So you're going to make the predictions from the single nearest neighbor. Whatever the closest point to me is, that's what color I say my new sample is. Okay, so that's the function in R to do this is KNN, for K nearest neighbors. You have to specify K, which is over here at K equals 1. And this is the picture you get out of K nearest neighbors. Now, this should get almost in fact, get every single training data point right. Why? Well, you only get one vote. You look at the nearest neighbor to the point you're trying to predict. If you're trying to predict on somebody who's in the set, that's yourself. You say, what color am I? Oh, I'm orange. That's what I'm going to predict. So this will get everything right on the training data, but that doesn't necessarily mean that it will get everything right on test data. And we can sort of see that, right? You can see the shape of the boundaries between this. There's no nice clean line. There's no nice smooth curve. There's a whole bunch of little islands in the river this time. It gets some of the structure right, but it has probably overfit the data. That's the problem with classifiers in general. If you give them too much freedom, they will overfit the model and make it more complex. So there's more complexity here than we know in the true model. Because it's trying to get every little local place right. Okay? And so actually, if you look here, right, on the training data, it's perfect. It's 100% accurate, it has 100% sensitivity and specificity. That clearly can't be true. We could, as we know from what we expect from the true model, it's overfitting the data. So maybe we can do better if we ask 5 neighbors to vote instead of just 1. Okay? And that's a little bit better, right? It doesn't get everything right now. There's a chance for having more neighbors of the other color even on the training set than just what you are. It gets this sort of bulge out here, more or less correct. It gets an island over here. It's missing the stuff from the true model. Remember, there was a batch of orange down here and up here that's the far bank of the river, right? So it doesn't get that, which actually isn't terribly surprising after all. It's really hard to extrapolate out to regions where you have no data. Right? We didn't have any actual data there. We only knew from the theoretical model what color that far side of the river should be. So in many cases, it will work that extrapolating outside the region where you actually have data is always problematic. It's really hard to get that correct. That's one of the reasons that sample sizes to do classifiers get bumped up so high. You have to make sure you have enough samples to get an adequate description of where the data actually lives. Too few samples, there's whole regions where you have no data, and when you get a new sample in there in the future in that region, you have no idea how to predict it. You're likely to be wrong on predicting stuff outside the region where you've actually seen data. Okay? And anyway, with k equals 5, we see that we don't get perfect answers on the training set. We get about 85% of them right. And we have about the same sensitivity and specificity here. So we're just as likely to get blue ones or orange ones wrong. And if we look at k equals 11, well, at k equals 11, some of the structure has gone away, right? The islands are all gone. This bulge in here remains, but the island that was over there is gone. We don't have anything on the far side of the river. And if we look at how it works overall, well, it's still, in terms of accuracy on the training data, it's not any better than the k equals 5 model. It might even be slightly worse, right? It's 84% instead of 85%. And there's a slight bias towards getting, and I forgot which is blue and orange and A and B here, right? One of them only gets 80% right, the other gets closer to 90% right. So there's an asymmetry there in the predictions. One generalization of linear discriminant analysis is called quadratic discriminant analysis. Here the idea is you're going to draw a quadratic curve as the boundary. That turns out to be a consequence of some of the arithmetic and algebra that goes into working out the model. The statistical assumption that's different that I've sloughed over is that linear discriminant analysis assumes that the two groups have the same covariance matrix. And quadratic discriminant analysis allows for different covariance matrices in the two groups. And that's why when you do the equalities, the x squared plus y squared term doesn't cancel out, and you get a quadratic curve instead of a linear curve. And you see that reflected here, right? This boundary is a degree 2 curve. It's part of a hyperbola. That's the other side of the hyperbola. So actually, this model gets the little orange part over here in spite of not having the data. The bulge that was specifically carefully fit with the K-nearest neighbors is a lot smoothed out here because of this global model of being quadratic. If we look at how well the predictions work, we get on the order of 73% right on the training data. So far, the k equals 5 nearest neighbor is the winner on the training data without necessarily overfitting too much. Okay. I'm trying to decide if I'm going to tell you anything about how support vector machines work. It's this very nice idea. There's a sense in which it generalizes this notion of fitting quadratic curves or linear curves, and it does this by, oh, sure. Once upon a time, I used to be an algebraic geometer. There's a tool in there known as the Segre embedding, which comes down to, instead of looking at all linear terms in a polynomial, it either looks at all quadratic terms, all cubic terms. It picks some degree and embeds your data into a bigger space. And then it draws lines in that space, which corresponds either to quadratic curves or cubic curves or quartic curves or quintic curves. How many of those can I remember? I don't know. In any event, that's the underlying idea here in support vector machines, although it's buried in most of the descriptions. We're just going to look at what it does here. So that's the support vector machine fit to the data. We don't know what degree this is because it's, in effect, some arbitrary degree built inside the model. And it gives a much more complicated boundary, okay, which gets some features right. Again, there's the part over here where there's no data, but it still detects orange. It seems to think that our river is just a lake because it ends up here now. It gets this bulge, which is part of the features of the data. And it gets 82% right on the training data and is likely to do reasonably well on the test data. It's interesting in some ways that support vector machines are applied in an omics context. It's actually one of the few methods where there's a mathematical theorem that describes when it should work. You can go back to sort of the original papers in the original book. And what they basically say is that you need thousands of training points to get the models right. And the example that they give as their first application of support vector machines in the early publications on it is that the US Post Office has this training data set of handwritten digits. Because, of course, they have machines that scan envelopes, find the zip code, automatically tell what it is, and sort things out so they know where to send them, rather than have a human actually look and say, that's 43210. Okay? And that training set has on the order of 10,000 examples of handwritten digits. And support vector machines work really well to identify handwritten digits from the training set and the test set. And that's sort of the order on which they expect to be able to train stuff. In most applications in omics, they have 100 samples they're trying to learn stuff from. There's no reason theoretically why support vector machines should work, and yet they often appear to. So there's this unreasonable effectiveness of this method in applications to real data. Okay. All right, so now let's look at yet another method. So again, we're trying to look across a broad spectrum of different ways to try to classify stuff. This one has two different names. In R, it tends to be called recursive partitioning. It's also known as classification and regression trees, or CART, which tends not to be used because the person who developed that originally trademarked CART. And so people don't use it, and the R implementation is called RPart so that they don't infringe the trademark on CART. Whether you should be able to trademark that is a different discussion, which we won't get into. Okay? So the idea is that you're going to build a decision tree. And at each step, you're going to use just one variable. You're going to say, out of all the variables I have to predict stuff, I start by saying, if I split variable x at 1.2379, stuff higher than that, stuff lower than that, I get a good I get the sort of best first step separation. And then for each of those nodes at the next step in the tree, I split it again. I pick a different variable. Maybe I pick y, maybe I pick z, maybe I pick whatever the next variable is called. So in essence, you have to look at all possible splits of all possible variables to figure out which does the best job of splitting the data. Whenever somebody uses a word like best, you should immediately say, wait, how are you defining best? What makes it the best? And the answer in this case is that, as you can see here, there's some measure of impurity. So impurity is a measure of, if I have a collection of objects, how intermixed is it between the two groups I'm trying to separate? So if I have equal amounts of group A and B, that's as impure as it gets, right? So I should have some function that has a peak at 50%. And if I have all of one group, that's as pure as it gets. So if I have all A's or all B's, I have 0 or 1 somehow for the concentration of one of them, that's pure. So I want functions that are peaked in the middle and go down. There are a couple of functions that are often used for that. One is called the information index, minus p times log p. P is the proportion of A's in there. The other is called the Gini index, which is just p times 1 minus p. Both of those are peaked at a half and have minima down at 0 and 1. And so what you do is you're going to pick the split at each step that gives you the biggest change in impurity. So it makes the net nodes more pure than when you had them lumped together. And when you get them all completely pure, you're done. You can run the algorithms to stop earlier than that. There are criteria. As long as the total purity isn't worse than something, you'll stop. And so we're going to produce a tree. The function to do that is rpart. All of these models look similar, right? To fit the model, I call rpart. I have the same formula. Category is a function of the explanatory variables. I'm using my simulation data. You can show, so this is going to produce a tree, as I said. The rpart plot library here is just so I can see what the model actually looks like in terms of the tree to get an idea of what it's doing. Right? So when we start, right, up here, we have 49% group A's, right? And we're going to split on whether the x variable is bigger than or smaller than minus 1.4, right? And in this case, we get something that's awfully pure, and we're going to call that group B. And in the other case, we get something that's still pretty impure. And then we're going to split that again. This time we're going to split on the y variable. And we either get this group that's impure, or we get a group over here that's almost all A's. And so we'll just call those A's. And then we split on x, and we get a pure thing and another thing to split. So there's this whole tree that you go down, right? You make a decision on each variable at each time. So the way you make your predictions is you just work your way down this tree. And this is the picture we get here. So what's the notable feature of the boundary between the orange and the blue spaces? If you wanted to describe the shape of that, how would you do it? Well, there are boxes. And the reason there are boxes to some extent is that you're picking one variable at a time, right? So you're either drawing it x, so that's a vertical line, x bigger or less than something, or y, which is a horizontal line. So your boundary is made up of some combinations of line segments that are either horizontal or vertical. So you're trying to approximate whatever the shape is by that. And you can succeed at some of that. You can see that, the reason these boxes extend out here, that's the best you can do to capture that bulge in the real data if you're only allowed to draw horizontal and vertical line segments. And you actually see part of an island there that's this separate rectangle that comes in. Okay? So obviously we're not going to get perfect data for some curvy, complicated boundary when all we get to do is draw horizontal or vertical lines. But we can start getting there. And if we look at the overall performance, we're actually doing quite well. We've got just about 85% accuracy on the training data again. But again, if you look at the shape, it is capturing some of the essential features of the data that you would want it to capture to work on new data. Now, a problem with CART is that it builds one tree. Right? You get one choice. At each point, you make a split. You can't change your mind. If your early choice turned out to be a lousy one, you can't fix it. And so the obvious way to do that is to not grow one tree, but to grow an entire forest. And maybe there's an intermediate step of an orchard, but nobody's done that part yet. Okay? And so there's a tool called random forests, which randomly uses a subset of the data each time to build a different classification tree, and then it makes its predictions by averaging over all the trees in the forest that it builds. Okay? So, not surprisingly, this is clearly not a Unix developer, because they would have just called the function rf. They actually call it random forest. Okay? So if we see what it does, there's the boundaries from random forest. Again, they're not quite just vertical and horizontal lines because it's averaging over stuff, but there's clearly aspects of that that are here. Do we think this is underfitting or overfitting the data? I think it's pretty clearly overfitting. You can see some of that in the same ways that nearest neighbors with k equals 1 was overfitting, right? It's gone out of the way to find this little piece here and this little piece here. Now that may be an aspect of the fact that we only have a two-dimensional data set. I have heard people claim to me that this works better when you have lots of variables to split on, and this randomization process works better. I mean, part of it is the randomization. I have to use variables x and y. I don't have anything else. If I have 10,000 variables, I can use different combinations of them and get some more flexibility that is maybe not likely to overfit the data. But here it's pretty clear that it's doing more than it should. Let's see. It gets perfect predictions on the training data. If you get perfect predictions on the training data, that's always a clear signal that you have almost certainly overfit something. There's two reasons for that. I mean, one is predictions are hard, and so you shouldn't expect to be able to get it perfectly right. There's another more subtle difference that's worth keeping in mind, which is that if we start going to predict like real outcomes that we care about from clinical data, and you say, I'm going to learn who responded and who didn't respond to treatment with this drug. That's the kind of thing we want to do ultimately. What you have to realize is that the data you're training on, who responded and who didn't, has mistakes in it. So the outcomes you're trying to predict, any clinical data that's been collected by some undergraduate sent to read the databases and record stuff, or some research nurse sent to do it, or some postdoctoral fellow sent to read it and do it, they make transcription errors. They type the wrong thing. Any real data, the labels you're trying to predict, include some that are wrong. In that case, if you learn them perfectly, you have overfit the data. You will make mistakes on the new data. So you need some method that is sufficiently robust that it doesn't get everything right on the training data in general. Okay? Now, it is interesting here because what's nice about random forest is because it uses a subset of the data each time. Right? It uses a random subset to learn it, so it gets different trees. It can make predictions on the ones that didn't get used on that model. These are usually called out-of-bag predictions or out-of-band predictions. And it will actually report in this random forest model how it thinks it would behave based on the out-of-bag predictions. And so you can actually look at this. The actual predictions it makes on the training data appear to be perfect. But if you use the out-of-bag predictions to see, it actually says it's likely to only be 77% accurate on new data, which is probably a realistic estimate. So it has estimated that by actually learning from a subset of the data and predicting on the rest and keeping track of how that works over many splits. That's a general tool called cross-validation that we're going to be interested in using in other contexts as well. Okay. This is a method known as naive Bayes. So here the idea is you pretend that all of the variables you have are independent. Each one you get some probability estimate, and you combine those probability estimates from all of the variables to see what happens. Turns out that in naive Bayes, it gets this bulge, but in this particular example, it gets it by some nice smooth curve that isn't very complicated. It gets that one salient feature of the data, but doesn't get much else. There's a tool called neural networks. I'm just going to skip through some of these. That's the neural networks prediction. Neural networks has the challenge that you don't really understand why it's making predictions. All right? So it's fitting some black box model that's hard to interpret. People like things like CART because you can say exactly why you're making the prediction. You know which variables it's picking at each step, which ones are big or not. If those are genes, you can actually give, biological interpretations to why you're doing that. With neural networks, it's just this black box. It makes predictions, but you don't know why. But it often makes accurate predictions. It gets some interesting shapes here. Here's the bulge with, I don't know, a little bit of spin-off of the river here and some triangular island over there. And that's an approximation to some of the real features. So it's not doing terribly. Again, I don't think I actually did the training data here to see what it does. There's one more method called compound covariate predictors, which again turns out to give a line, and we're not going to focus on that at the moment. What we're going to try to look at briefly is how this is likely to work in an omics setting, now that we've sort of seen some of the behavior of this in that two-dimensional case where it's hard to do. So I'm going to load a data set called Classify. We're going to go through this part a little bit quickly so that we have time to go through the continuation of the breast cancer data set that we looked at yesterday. So what do we have? We have training data that has 12,000 genes in it and 198 samples. It has a simplified clinical component with it where there's obviously some subtype of cancer, which looks like there's 6 different subtypes there. There's an outcome that's just labeled as bad or good. There's a last follow-up time, and there's an event. So there's probably a survival data here. The event is whether this is censored or not. We've talked a little about survival data earlier. And if we look at what the actual data looks like, here's a box plot. So it's clearly on a log scale of some sort. It looks like it's been normalized reasonably so that the individual samples look like they have similar distributions. And here's a plot of the actual outcome data in the first two principal components, which says this is not going to be an easy thing to predict. Right? The good and bad outcomes look like they are smeared randomly and equivalently across the first two principal components. So maybe there's other principal components, or maybe there's a small subset of the genes that will separate them that is not the major source of variation here. So this is challenging in a different way. We can't see in the principal component space, at least the first two principal components, how to tell good from bad outcomes. Outcomes tend to be harder to predict because, in part, it may, well, there are a couple of things. One is that it may just be that it is a small part of the gene expression pattern that explains the outcome. Right? If you're trying to treat with some drug, that drug targets a single pathway, maybe it's only the expression of the genes in that pathway that are relevant, and the other 20,000 genes are irrelevant to whether you respond or not. And so if you look at the overall pattern, you don't see it. You really do have to work hard to pick out the features that can be used to predict things. So I'm going to, again, as I said, go through this a little on the fast side. There's a package called Modeler, which we had to install the other day. There's two more R scripts here that add some additional features to that. But the idea, what I want to look at, is that there are notions of filtering genes and feature selection. And I want to make a distinction between those. We talked about this to some extent in the clustering issues yesterday. Right? I don't want to filter things to discover structure based on the outcome that I want to predict. Right? So filtering, I'm reserving for the idea of I pick things like mean expression, standard deviation, coefficient of variation, some other sort of feature that isn't related to the outcome, and I reduce the complexity of the data set by throwing away genes based on they're all low expressed, they're not very variable, they're boring for some reason. Feature selection, on the other hand, is an essential part of picking the classifier. In this omics context, where I don't have just two variables, x and y, I have 20,000 genes. Picking which ones I want to use for the model is a key part of it. That's feature selection. So that's picking things based on how they relate to the outcome. And there's a whole bunch of ways to filter things, that is, using measures other than outcome to get rid of genes. You can do it based on mean expression, median expression, range of expression, whatever the 75th percentile is, whether there's a gap in the data so that it suggests that it separates two groups, what the minimum expression is. IQR is the interquartile range. That's what's shown on a box plot from the 25th to the 75th percentile. The standard deviation, whether there's some evidence of bimodality. And so we can look at applying all of those filtering methods and ask, for example, how many genes survive different filters. So again, we started with 12,000 genes. Depending on which of these filters I want, mean and median, at least with the levels that I set, I still have about 9,000. Some of these I have 8,000. Whether there's a gap or not, that's sort of rare. That gets me down to 3,000 genes. That might be interesting, right? If there's a big gap in the data that suggests there's two groups, and if I want to predict a binary outcome, some of those things might help. Similarly with bimodality, which is maybe more severe than gap, right? You're looking for whether the data has a distribution that looks like that, which again suggests that there's a high expressing and a low expressing group. That's always a signal of something. Whether it's a signal of the outcome you want to predict, you don't know. But if you're looking for predictors, that's a better candidate than something that's just normally distributed in just one group. So there's various ways to do this. But the point in part is that you get very different answers depending on which of these you use, and you can combine them, right? And if you ask something that meets all these criteria, how many genes are there? In this case, there's only 200. So you could filter way down if you want. If you filter down too much, you may lose the predictors of interest, however. There's also a gazillion feature selection methods. Again, feature selection is I'm going to pick stuff because it appears to be related to the outcome. And my first choice is to keep everything, right? Not do any feature selection. That's going to be rare. We could pick things based on a t-test. I know the two outcome groups. This is one of the more common things. I do a t-test between them to see if it's differentially expressed, and only keep genes that are differentially expressed between the two groups to try to predict in some formal model later on. You can also do that with a chi-squared test by making things discrete. You can use Pearson or Spearman correlation to see whether the expression of your gene is associated with the categories that you want. You can use various measures that come from information theory. You can use a tool that's based on random forests. What I didn't tell you about random forests earlier is one of the other pieces of information you get at, because again, you've split the data many times, is how often does a gene get picked inside one of the trees that the random forest gets. And the more often it gets picked, the more likely it is to be useful as a predictor for that particular outcome. So you can use that as feature selection as well. And again, I want to make the point that different criteria, again, give you very different numbers. Some of them, in this particular data set, very few things pass the t-test criterion. Many more pass the correlation criteria or the information theory criteria. So again, the whole point is that there's a whole bunch of ways to do this. It's going to matter which method you use. And then we've got all of these classifiers. Right? We've already talked about, nearest neighbors, CART, random forests, support vector machines, naive Bayes, neural networks, maybe a couple of others that I haven't talked about. But you can, so I actually ran this code to pick a filtering method, pick a feature selection method, pick a classifier, apply it, and see what happens. Now, part of this is I made this for a class originally, and you could do this, because if you run this with a different seed, or without setting a random seed, then you end up picking different random combinations of this, and you can test how different filtering methods, feature selection methods, and classifiers work on the data. And so that's the point in part that this isn't done automatically. It's there so that if you want to go play with it, you can randomly run this and see which ones work best on the data set. Now, rather than do that, we're going to take a deep breath and go to by the way, the reason I have to keep looking over there is the monitor here hasn't turned on, so I don't actually know what I'm projecting there. And so if I have to move the mouse, I have to see it over here. So we're going to close this down and go back to our hands-on part, which I told you to get yesterday because it has the combination of stuff from both the clustering part and the hands-on part. So the HTML part is here, but this time we're going to switch to doing this in RStudio. And try to do this in real time, a chunk at a time. And oh, I don't like when it puts the output in there. I like when it puts the output in the console. So yeah, you can remove the output, and I'll do this again. Okay. So I need some libraries. And I need all the data that I saved from the clustering part yesterday. So we should be sort of where we were yesterday, right? So everybody remembers, we were looking at breast cancer data, and from the clustering point of view, we were trying to say, how many clusters are there? There was a red cluster that was the most robust cluster we could find, which we sort of guessed at the end of the day represented triple-negative breast cancer. There was a small purple cluster, that was the second most reproducible thing that split off, which we thought was probably HER2 amplified breast cancers. And then there were 2 clusters that were labeled blue and green that looked like they were both hormone receptor positive. And the question was whether we could actually tell them apart reliably enough to believe they were 2 groups. So that's the review of where we were as we finished the day yesterday. So what am I going to do? Well, first of all, I'm going to convince you again that these really are groups that are related to ER and PR and HER2. So this next chunk just picks out the data for the estrogen receptor, whose official symbol again is ESR1, the progesterone receptor, whose official symbol is PGR, and HER2, whose official symbol is ERBB2, because the Human Gene Nomenclature Committee doesn't like bioinformaticians. Okay, so let's do this plot first. So this is a histogram of the expression of the estrogen receptor in this data. My discussion of bimodality, if you were filtering this data on bimodality, ER would be way up on the list. There's clearly 2 different modes, right? There's a small group where the expression is around 6 on a log scale, and a big group where the expression is around 13 or 14 on the log scale. And you can, by eye or more formally, draw that blue line at some place that more or less divides the high-expressing ERs from the low-expressing ERs. That's the cutoff that I used arbitrarily near the end of the day yesterday. Okay, so certainly the estrogen receptor has this bimodal distribution. Okay. Now the progesterone receptor also has a bimodal distribution, but it's not as well separated as ER. But there's clearly 2 lumps to it, a higher group and a lower group. They're not as well separated as estrogen. But again, I can make a high PR and a low PR group. And we can do this for HER2, where there's a much smaller, well, actually it looks like it's trimodal. There's this really big low-expressing group, where low-expressing, by the way, is like 12 on the log scale. So even low-expressing HER2 is pretty high. Right? And then there's 2 further groups, which I guess is amplified and even more amplified, which we're just going to count together. Okay? So I'm going to say that the things above this line are the ones I think are particularly high HER2. It would be nice if we actually had data from the way ER and PR and HER2 are measured in clinical samples. Right? It's done typically by immunohistochemistry on the samples to look at proteins. All we have is gene expression. We don't have protein expression, so we're just trying to approximate it from this data. Okay. Yeah, let's see. This is a box plot of expression of ER by the colored groups that we had from the clustering. So we're using the same colors. So what do we see here? We see that the red and purple groups are low for ER and the blue and green groups are high. That's what we expected from what we looked at last time. The box plot just gives a clearer idea of that. We can do the same thing for PR. It's low in the red and purple groups, higher in the blue and green groups. And we can do the same thing for HER2. Which is clearly higher, at least on average, in the purple group, although there's some of the blue ones that are high. Right? And it's relatively low in the red, blue, and green. So this is what gives us the interpretations that we had yesterday. Right? The red is low for everything. It's triple negative. The purple's low for ER and PR, but high for HER2. It's the HER2 amplified. And the blue and green are high in ER and PR. They're the hormone receptor positive. Okay? So that just recapitulates what we had before. I want to do these. You can also look at the connections between these. So this is a scatter plot of ER versus PR colored by our clusters. We're not doing any classification yet. We're still making sure we understand the data, right? And it's sort of what we expect, Right? Again, the blue and green are sort of smeared together on this in that ER and PR mostly track one another, but there's a lot of variability between them, of course. That's what happens when we actually look at this data. You can see the trend of going up to the right for both of them. They're correlated, but there's still a lot of noise. We could also compare, maybe we'll skip to here, right? ER and HER2 are not particularly correlated, but you can see the bimodality in each. If you look carefully, right, there's the blue-green group on the right, the big group, that's high ER and low HER2. There's the red group at the lower left that's low in both. There's the purple group above it that's high in HER2 and low in ER. And there's a few outlier blues primarily that are high in HER2 even though they're ER-PR positive. Okay. All right, so now we're going to do classification, right? We want to distinguish blue from green, right? Right now we don't have a better name for these cases of breast cancer other than we, by chance, picked blue and green to color them early on, okay? So, and now I want to look at this scatter plot. So there's a little bit to mention here. You get to remember silhouette width. Silhouette width, remember, is a measure of how well classified we think a sample is when we put it into clusters. And we're looking at 4 groups here because that's how many we think we should be looking at. Red, purple, blue, and green. Okay? And remember the interpretation of silhouette width. Closer to 1 is well classified. Closer to -1 is terrible. And around 0 is probably not that good either. Okay? So if I look at this picture, what do I see? Well, again, the red ones are well classified. We understand triple negative. The purple ones are okay. I mean, they're mostly positive. There's 1 or 2 that's negative. But not surprisingly, for the blues and greens, we run into this problem because the blue and green overlap. There's some of them that we're confident are blue, some that we're confident are green, and then a bunch that we're not sure about, but mostly we're not sure about because we don't know which of these groups they belong to. So I'm going to do something unusual in the role of trying to build a classifier. I'm going to pick the blue and green ones that I'm confident should be blue and green, and not try to learn the model, including the ones that I don't know how to classify. This tells me I don't know how to classify them. Trying to learn a model from those is probably a dumb thing to do. But most people don't bother to do this step first. Okay? So let's run. So we're going to pick, as I said, the blue and green samples that are well classified. And out of the 1,000 samples we had to start with, we're throwing out the reds and the purples and the hard to classifies. We're going to keep 550 something samples to try to learn a classifier, which might even be enough to do something. Okay. And how do they break down? Well, they break down with, of those, we have 403 blue ones and 151 green ones. Okay? They're just more blues than greens. Whatever that subtype is, it appears to be more common. And now I'm going to split that group into training and test set. Because we know, or at least we should know because I've told you a couple of times, that whatever we learn, we want to be able to test on something that wasn't used to train the model. So I'm going to split it into training and test. And so blue and green, and there's this true means I'm going to keep it for training. False means I'm going to leave it for test. Okay? So that's just a matter of how many I have in each group. So I have 184 samples that I've put aside to use for testing whatever models I built. And this actually does the splits and gives them names that we can use. And we're going to use the class comparison package for something because the sort of feature selection we want to do is going to use gene by gene t-tests. Right? That's one of the feature selection methods I described as probably the most common one. So from these robustly classified blue and green samples, I'm fitting t-test models to each gene to see what happens. Okay? And I want to see what that distribution of p-values is from the t-test. So, do we know how to interpret this histogram of p-values? What does that tell me? Well, if there's no difference between the blue and green groups, my histogram of p-values should just be flat. And if I have an excess of p-values near 0, there's a spike. So which of those cases am I in? Oh, come on, you had some coffee already. You can't be asleep. Yeah. Clearly, there's this enormous spike at 0. You can barely see the flat part. A huge number of these genes are differentially expressed between the robustly classified blue and green groups. It's not just a few genes that are driving this. Most of the genes are different. That's really strong evidence that these are really different kinds of samples. Right? Almost anything I do, this says I should be able to tell them apart. Because if I ask how many there are at a given false discovery rate, right, at a false discovery rate of 0.1%, I'm getting 7,800 genes that are different between blue and green. And 0.1% of that is, so maybe 70 of them are wrong. There's at least 7,000 genes that I should really believe are different. It's almost impossibly different. Okay? There is no way these are the same kind of samples. Even at setting the false discovery rate at 10 to the -6th, which is so small that I'm using scientific notation to do it because I can't trust myself to type that many zeros correctly, I still get almost 5,000 genes that are different at a level where none of them are expected to be false positives. Okay? And after filtering the way I want to filter, well, I still have 500 of them at this ridiculously tiny false discovery rate. Of the ones that would pass some filtering criterion based on expression. Okay. This filtering criteria, by the way, is what we defined yesterday. Remember, we set a cutoff on the mean expression and a relative cutoff on the standard deviation. We had about 1,000 genes that were past that filtering when we did the clustering. Here I've done the t-test on all genes, not just the filtered ones. But that 481, that's almost half of the genes that I kept that are still differentially expressed between the groups. Okay, so let's see. I'm going to save that list because there's a talk later on using ingenuity to understand gene sets. And that list of luminal genes at least was the one that Molly planned to use. We'll see if she's still using that data set. But that's where it came from. We're going to look at this filtered data set. So those are my filtered genes, my 550 robust samples. Okay. And now we're going to load our modeler package. And we're going to actually look at, for example, in this case, KNN with n equals 3. And how does it do? Well, we're going to look both at the training and the test sets. Right? So I learned from the training set. Notice that it gets 6 wrong on the training. Oh, train equals false. This is the test set. This is the training set. So on the test set of, let's see, 134, of roughly 180, 190 samples, only 6 of the samples on the test set are predicted incorrectly. And again, the models fit on the training set. There's only 7 that are wrong there. When they're wrong in the same way. This is truth across here. These are predictions this way. In this case, we do about as well on the test set as we did on the training set using KNearest Neighbors. Question?
Why is A equal to 3? How did you pick
Because I don't know why I picked 3 here. We could try a different one. That's one of the reasons of having code here that we can test. So, because I had predefined, so part of it is in the modeler code, 3 nearest neighbors is one of the things that they had predefined as a model to use. So I didn't play with K here. I just tried 3 nearest neighbors to see how it works. Because I never trust 1. 3 is the sort of next thing where at least there's a chance that I don't overfit the training data. But this says I didn't overfit the training data. Right? The results on the test data tells me whether I've overfit. Okay? So we can look at CART and see how well that does. There's the training data results. There's the test. They should be in the other order. It's because of this true-false that it sorts automatically. That I keep. There's the training data results. We get 6 wrong out of however many. We get 14 wrong here. But still, the accuracy of the CART model on this data set is very high, even when I look at the test data that was not being used to train the samples. Okay. Compound covariate predictors, the results are similar. We get 5 wrong on the test data. Support vector machines. I mentioned the unreasonable effectiveness of support vector machines. It gets nothing wrong on this training data set. It gets nothing wrong on the test data. There is no theoretical reason it should do that well. Now, part of this turns out to be an easy classification problem. It's easy because these are really different groups. But it's also easy because I focused on things that I thought were easy to classify. That step where I said, I'm using the silhouette width, I'm going to learn my model on things that are clearly green or clearly blue, and not try to fit the model so it accounts for the ones that are blurry on the borderline, because I don't know what to call those. Those are the ones that are ambiguous. Each of these models will make different predictions on those. And to some extent, given how well they're working here, it's the predictions they make on those hard to classify blues and greens that probably tell you what group it should really belong to. Random forests. Takes a little longer to fit random forests because it has to build a lot of trees, and you have to plant seeds and wait for them to grow and harvest things and do something to fill in time while it runs. So random forests is perfect on the training data, but its error rate on the test data is similar to other methods. So it may have overfit a tiny amount, but this is such an easy classification problem, it turns out not to matter. We can do linear discriminant analysis because we have fewer genes now having filtered than we do samples. It goes very fast. It's also perfect on the training data. So in fact, there's a straight line in some dimension that separates things. It gets a few more wrong than some of the others. There's 16 errors here. What else? So cross-validation. So I've again done this with, this is actually time-consuming because I'm going to split the data 2/3 for training, 1/3 for test. I'm going to do it 50 times. I'm going to fit a bunch of models. So I cross-validated some of these and saved the results, and uh-oh, that looks bad. I forgot to install cross-validate. Okay, let's see if we can fix that.
Yeah, don't attempt to do it if you don't have the software.
Sorry? Enter. Okay, so now we wait while it cross-validates, and then we see if I can get this to work. Okay, let's try again. All right. No, how about I click here? All right, so now it doesn't moan at me, so we can look at the results. So I should probably fix this so I can see the boundary here. All right, so this is cross-validating the support vector machines method. Okay? Every time we split it, it's perfect on the training data. So it can always completely predict what happens there. And it actually has very good accuracy on the validation data. Right? The overall accuracy ranges from 98.9% to 100%, with similar ranges on the sensitivity, specificity, positive predictive value, and negative predictive value. Support vector machines just works on this data set. K-nearest neighbors, if we look at that, well, it's not as good, right? Instead of 98% to 100%, well, the accuracy is 96 to 99, gets lower levels. The specificity tends to be higher than the sensitivity. But if I look at the validation set, again, it's lower, right? I mean, we're looking at about 95% accuracy instead of 99% accuracy on average. The compound covariate predictors method, well, it's probably a little better than K-nearest neighbors. It's not quite as good as support vector machines. The linear discriminant analysis, again, it can always perfectly fit the training data, but if you look at the performance on the test data, it's not nearly as good as support vector machines. Right? We get a median accuracy of only 89%. On the sensitivity, in at least one case, it's as low as 60%. So even though, in some sense, this is an easy prediction problem, much easier than I expected when I first started it, because we saw all that blurring between the blues and greens. In this particular case, linear discriminant analysis has a tendency to overfit, and so it doesn't work as well on new data. So the takeaway for this particular data set is that support vector machines is the classifier of choice. I can guarantee you that if we look at enough different data sets, that conclusion will change. But it is a reasonable one to try. Maybe it's an unreasonable one to try, because as I said, I don't understand theoretically why it works as well as it does in so many cases. So let's stop there and give you a chance to ask some questions.